Hello, everyone, and welcome to the Use of Microbial Fungicides in Specialty Agriculture, Lessons from the Field webinar presented by BioWorks. This discussion will showcase real-world results of how biofungicides boost yield and health in specialty crops. I'm Hillary Wynn, the Custom Content Manager for Meister Media Worldwide, publisher of American Vegetable Grower magazine, and I'll be moderating this webinar. This webinar has been approved for 0.5 CEU credit in Integrated Pest Management from CCA. To receive credit, you must watch the entire webinar. An attendance notification will be sent at some point during this webinar, and you must respond to receive credit. If you respond once the webinar is finished, you will receive an email with the QR code for reporting your credit. You simply scan the QR code and submit it. Please do not close your web browser before the webinar has ended completely, or you will not receive the email with the QR code. This is also for the viewers watching the archived webinar. Now, I'd like to go ahead and introduce our speaker, Dr. Eli Blanco Rodriguez, Technical Services Manager for BioWorks. Eli holds a PhD in Agronomy and Crop Science from the Agricultural University of Havana. He is an accomplished agronomist with over 15 years of expertise in plant protection, specializing in biological control and integrated pest management for both indoor and outdoor crops. Eli has authored more than 40 publications in the fields of entomology, IPM, biological control, insect systematics, and pest risk analysis. At this point, I will turn the webinar over to our speaker. Eli, welcome.
Thank you. Thank you for the presentation. Let me just pull out the slides here and just let me know if everything is okay on your side. Let me know. Is that okay?
Yep, we are good.
Perfect. Today we are going to concentrate on the use of microbial biofungicides in particular in specialty crops. As Hillary mentioned, we're going to concentrate on lessons we have learned from the field in implementation of our strategies. Particularly, we're going to be talking about 3 crops in specific: lettuce, celery, and strawberries. Those 3 crops are, really affected by Fusarium complex. And as an introduction, we can say that Fusarium wilt complex is a devastating group of soil-borne diseases that can cause severe damage in all of these crops. Over the last 20 years or so, Fusarium wilt have become a common phytosanitary problem in many other crops. And just to mention a few, we can concentrate on tomatoes, watermelons, onions. All of those have, their specific forma special species that affect them in particular. Over those years that I mentioned as well, we can mention that over 100 host plants have been reported for the Fusarium complex in general. And disease management is basically concentrated on the use of the genetic material that offers, certain level of tolerance to the disease. By saying that, I have to emphasize that chemical control is very limited in all those cases. Today, we're going to be speaking about a group of products. Some of them are included in our portfolio. This is one of them. This is one of our most important products in BioWorks portfolio. Crucial Plus WP is a certified biofungicide. And basically, its mode of action, it's by competing. It contains basically 2 strains of Trichoderma harzianum T22 and Trichoderma virus G41. All of those target specifically Fusarium complex in particular. T virus in particular is very efficacious against oomycetes in particular. And as I said before, the mode of action of this product in particular is by competing with the pathogens that might be present in the rhizosphere. Fusarium is included among those species that our product is very efficacious against. It also shields basically the roots of the plants, offering a certain protection against those pathogens in the soil. It also hunts down the hyphae of those species of pathogenic fungi in the soil. Here we can show an image of Rhizoctonia hyphae being penetrated by the hyphae of Trichoderma in particular. It can also offer other more sophisticated modes of action, in particularly by releasing fungistatic substances in the rhizosphere, and by doing so, it limits the growth of those pathogens in particular. Among those sophisticated modes of actions as well, we have to mention that it triggers the natural defense mechanisms that are normally expressed in the plant. But in this case, it primes them and potentiates them to a higher extent, conferring more protection to the plant in its natural way. We're going to be speaking as well about the use of OnGuard, which is registered as a biofertilizer in particular. This product is considered a natural nitrogen source, in particular because of the content it has. It's based on protein hydrolysates with a very low sodium content. It increases the stress tolerance in the plant, specifically to extreme temperatures, to drought and salt content in the soil. It also enhances nutrient uptake and overall absorption. As you may know, amino acids or protein hydrolysates are chelating agents by nature, and by that means they carry most of those microelements into the plant in an easier way. It also activates plants' natural defense mechanisms by, potentiating those metabolic pathways in the plant and rendering it more prepared, so to say, for those both biotic and abiotic stresses that might be present in the field. It also promotes root growth. It's compatible, we have to emphasize that it's compatible with most fertilizers, chemicals, and biological products. In fact, one of the uses that you'll be seeing in the presentation, over the course of the presentation, is the use of these products as a carbon source for the products that we are going to be mentioning. So not only it by itself, it confers many benefits to the plants, but it also confers advantages to the microbial products you can use in the field. It can be either applied as a foliar spray, as a root drench, or through the irrigation system. I should have mentioned that both of those products are unreleased, by the way, so their use in organic production is very well welcome. Another product that we'll be mentioning during the course of the presentation is Chamay. This is not a product that is part of the BioWorks products, but as we have been using it in conjunction with our products, I will mention some of the uses we included in our trials, so to say. Basically, it's also a blend of essential amino acids to the plants, but it also contains humic and fulvic acids. It also contains 5 essential organic sugars, organic plant extracts, and it's reinforced with the presence of nitrogen, potassium, and phosphorus, and microelements which are not present in other biofertilizers or biostimulants that are normally encountered in commercial production. It is also improved in its formulation by the inclusion of nanocatalysts. These are nanoparticles that render more easy the assimilation of elements by the plant in particular. So jumping into the specific uses of our products, the first case study we're going to mention is the use of Root Plus, OnGuard, and Chamay. this is a strategy that we came up with as a result of the request from one of the growers in Ventura County. They were facing many problems with Fusarium oxysporum forma sociale API in celery in particular. As you may know, this disease was first detected in 2012 in particular. And there are no other means of control for it. So it's really a very aggressive disease with the crop. It can render the crop completely dead in a matter of 4 weeks, so to say. So on the left side, you see the first trials we were conducting in 2022 and how has that progressed in the years, over the course of the years until 2024, and even until the present year where we are implementing this strategy. So one of the tools that we have came across and defined as very useful for the growers is the use of Rootchill Plus, or in this case, T22HC, which is a formulation that we have in particular in our portfolio for seed inoculation. So in this particular slide, we wanted to just show to you how the inoculation of the seeds can be an useful tool for the growers, just to at least prime those plants prior to arriving to the field. One of the most important elements that the growers see when they transplant their seedlings in the field is a lot of transplant shock. So this could be an important element for them to reduce that transplant shock, and by doing so, reducing the possibility of those transplants to get infected with Fusarium API in particular. So as you can see on the right side, we have a graph in which we inoculated seeds of a very susceptible variety with T22, and basically T22HC contains Trichoderma halcyanum, just to be more specific in that case. And we work with a priming and coating company that offers their service in Salinas in particular. Normally, this company claims that their seeds should be used after 90 days, for example. And we were able to confirm that our product, once inoculated in those seeds, is able to comply with the technical elements they mentioned. And not only that, we're almost able to obtain very good inoculum levels even after 147 days, as you can see on the graph there. How does that translate in, the level of protection we see in the plants? Well, on the left side, you see plants that were, not inoculated on the right and left sides. And on the center, you see 2 rows that are specifically the plants that were obtained from those seeds that you see on the right graphs. So as you can see, the plants are able to tolerate the disease in some extent once they're inoculated with Trichoderma halcyanum. However, we proved that by doing so is not enough. You need to continue on with the strategy once you arrive in the field. And the reason why, although I didn't include any images here, is because those plants that you see in the center were not able to overcome the disease. And in fact, they succumbed to the disease after 4 weeks of being in the field. So, basically seed inoculation is not enough to protect the plants, and you need to reinforce this strategy with other elements that we'll be talking about later on. So another element that we need to include in the use of biologicals like this ones is the inoculation of the soil of those seedlings. So particularly by doing so, you will be able to, render those roots of the plants even stronger. The root biomass will be bigger. And as you can see on the right graph there, we were able to increase up to 10% the root biomass in the treated transplants when you compare them to the non-treated control. So that translates as well with a more vigorous plant, sturdier plant that is able to overcome those negative, effects that the pathogen confers to those plants once they arrive in the field. So basically, this slide here shows our first case study again in a commercial area. Just to give you some elements about it. Both of these field sections that we are going to describe as section 1 and section 2 were infected with Fusarium race 4 in previous crops. So this is an area with high pressure of the disease. And by the time the grower called us, 15% of the plants, of the seedling plants, were already affected by the pathogen. Both sections, as you can see there, contain around 30 acres each. They were treated with the 2 strategies comprising the use of Rutile plus WP plus Chamay as a combined application to the soil. And then we continue on with an application of On Guard. This was repeated on the left side on 2 occasions, and on the second side or second section in 3 occasions. So just to emphasize, Rutile Plus and Chamay were applied via drip tape, and On Guard was applied as a foliar treatment. Both were compared with the standard control that the grower had very in the proximity to these fields. And basically it contained all the conditions, same type of soil, same type of infection rate. And they were basically implementing what they normally do on their commercial conditions. We measured a number of variables. In this case, we were measuring the incidence of the disease. We also used NDVI, which is the Normalized Difference Vegetative Index, as an element to kind of monitor the health of the crop along the cycle. We were also monitoring the soil health, yields, and product compatibility as a complement to what the grower is normally using in terms of pesticides and herbicides. Oops, something happened here. So the first element we were able to collect through the crop cycle was, the NDVI, as I was mentioning. And we were monitoring the whole crop ever since it was transplanted until harvest. So as you can see, and I should probably emphasize that NDVI values are read between 0 and 1. So the higher the value, the closer to 1, the better in terms of health, crop health, we can read it. So for Rutile Plus, as you can see here, the first application of Rutile Plus was conducted here, and the readings we were obtaining from this application, which name is One Soil, by the way, is 0.77. And as you can see, as we progress with the applications of our products, we were able to see an increase in the values of NDVI that were subsequently registered by this application. And this is probably, a graphical view, but if we transfer that to an image, a satellite image, which we can see on the top right side over there, you see how the crop progressed very fast from being very yellow and with areas showing certain level of susceptibility to very green over the course of the crop. So that means that the crop was able not only to overcome the stress conditions by the disease in the field, but getting very healthy as the time progressed in the field. Another element that we normally monitor is soil health, and this is becoming a trend for many growers in specialty crop. We want to see an improvement in the microflora or rhizosphere consortiums, and particularly because of the consistent and continuous exploitation that the commercial fields has had. We see very low numbers of beneficials included. So by doing this type of strategies, what we are pursuing not only is the health of the crop, we are also pursuing the improvement of the biological characteristics of our soil. An improvement, basically the improvement of the soil consortium, and seeing better conditions for the plants to, cope with the negative effects that pathogens might have in the field. So basically, what we wanted to show in this graph is, the ranges in which the fungi bacteria biomass ratio will be registered. So during the first evaluation, as you can see, which we conducted on September 22nd of the year 2023, the levels that we registered for both sections treated here were rather low. According to Foothill Biological, which is a laboratory specialized in this type of analysis, the range in which these parameters should be included is between 0.3 and 0.8. Those are the best conditions for growing vegetables in particular. So by the second evaluation that we conducted in the field, particularly this was this soil sample was collected on November 8th, we saw an improvement of those two values in the two sections. And again, the optimal range for vegetable production was found after applying our strategy, meaning that there was a significant improvement in the soil consumption, and particularly the microbial consumption in that soil. So continuing on with the yields, which was another one of the parameters we were measuring in this particular case, we were able to accomplish very good results in terms of yields in both of the sections we treated. As you can see on the left side, the average yield per acre, and this is given in 55-pound cartons, was able to be bigger than the standard control that the grower was using in this case. If we use the line, the red line, as the level for the break-even point that the grower normally uses for, recover their investment, we can see that there's a 200 cartons on top of that 720 cartons that the grower was able to obtain in their standard control. But if we see the two sections in which we were implementing our strategy, we see an improvement of up to 4 times that revenue in terms of cartons in this case. So we were able to improve the number of cartons versus the control up to 4 times. So in a very summarized way. Well, not happy with that only, we wanted to see how would that stay in time. So this one time, it could be, a matter of possibilities that, and a matter of chance or good luck for us. So we wanted to see that our strategy was able to be consistent in time. So we implemented the same strategy in 2024, again, in susceptible fields. In this case, we were implementing it in 6 fields in particular, 6 commercial fields, all of them under commercial production. And as you can see on the left graph, on the left side graph over there, we were able again to improve the level of production that the grower was able to obtain on those commercial fields by implementing our strategy. versus the standard control. In this case, we obtain as an average 450 cartons per acre versus the standard control. In some cases, even more than that, if you see the variation that we included over there in our graph. Although this was not, in general terms, significantly, statistically significant, we can say that it was marginally statistically significant. So if you see the difference was set to 0.05, and we were able to obtain 0.07, which is very close in this case. And not only that, we were able to obtain, on average, 24 unit cartons increase in the case of our strategy, from 9 up to 17% when we treated, when we included our strategy in particular. One element that we have noticed over the course of the time is that the use of herbicides in commercial production is very frequent. The grower normally uses Caparol, which is the go-to product for them to combat many of the diseases that might be present in the field. And we wanted to know the impact of these herbicides on the 2 trichoderma species we were applying under field conditions. As you can see here, when we conducted our compatibility tests, this provided to us that none of those herbicides affected the trichoderma species. However, we can say that this is not a complete result, so to say. And I should probably stop myself a little bit here just to explain why. So if we see here, what we conduct in our laboratory is both a physical and biological compatibility test. And as you can see, the trichoderma species are able to grow in the presence of Caparol in particular, in this case. But when you combine it with OnGuard, which is a product that contains amino acids, as we mentioned before, we see some precipitation in the combination of these 2 products, which means it's able to denaturalize the substances, or in this case, the components that are present in this product. This tells me indirectly that we should be very cautious about, the use of these herbicides, particularly when it's very close to the plant in the field. Why is this? Because it can affect the photosystem too within the chloroplasts, and this interferes with the transport, the electron transport during photosynthesis. So that means that it's able to denaturalize, the exudates that the plant would be releasing for the benefits of those of that microflora that is present in the field. So all in all, what we have to take into account is that the use of herbicides should be as distant as possible from the use of our trichodermas in the field, because this can affect the performance of products containing trichoderma in particular, because it denaturalizes the exudates of the plants. Or if we mixed it with a product containing amino acids, we'll be observing the same problem. So the second case study that we included in our presentation is the use of BioWorks products, particularly for the control of Fusarium oxysporum forma speciale fragare. And in this case, we were working in organic production in particular. So as you can see here, we were implementing our strategy in Schuler, organic production. In this case, the grower wanted to test 3 strategies of our products, one of them containing Fusion, which is not a product from BioWorks, but is a product that the grower uses on a frequent basis. All these sections contain about 4 acres each, And we were comparing to a grower standard, which in this case contained a single-based trichoderma product. All the applications were conducted in this case via drip irrigation, so chemigation in particular. And we were measuring a number of variables, which I'll talk about later. Just as a reference of what we encounter, what we see in the soil, in particular that has been, conventionally treated for years. On the left side, you see that the microflora is very limited in those cases versus what we can observe in our granite production on the right side. This data is from Monterey County in California, by the way. So jumping into data in particular, what we were able to confirm by using our strategy is that all three strategies in particular were able to reduce the number of plants that were affected by the pathogens in this case. And as you can see, the combination of Root Plus at 2 pounds with Fusion at 1 pound and applied over the course of the crop was able to reduce even more the amount of infected plants, even at a significant level. And the same happened with the combination of Root Plus with On Guard at 32 fluid ounces. So basically, this is a very important element because by implementing this strategy, we are able to reduce the amount of plants that are affected by the pathogen, and that translates indirectly in more production for the grower. So not only that, when we compared our strategies with the growers, in this case, particularly one that contained Root Plus at 0.5 pounds and On Guard at 60 fluid ounces, we were able to obtain highly significant differences when evaluating all the yields that we obtained over the course of the year and over the course of the cycle of the crop in this case. And as you can see, we were able to obtain 1,200 trays more per acre compared to the grower standard in this case, which I should emphasize that was a single trichoderma based product. So basically 1,200 trays per acre means pretty much a significant increase in yields. And not only that, it also results in a very high revenue for the grower as compared to what they normally implement in the field. So the third case study we have for today was the use of Root Plus in combination with Chamay again. And in these cases, we have been using pretty much what the grower wants to use. And we also combined that with a foliar application of On Guard in particular. So the only difference here was that this was in lettuce, and we were trying to combat Fusarium oxysporum form speciale lactuque. This was conducted in April 2024, and the application, as you can see here, was through soil via chiseling in particular. Again, this was conducted in commercial conditions. The areas of both the BioWorks strategy and the grower standard were approximately 3 acres each. And we were measuring pretty much, the number of plants that were affected by the pathogen and yields as well. So basically, when we evaluate the head counts over here, we see a significant difference versus the grower standard by using BioWorks strategy. Particularly, we conducted an evaluation of the head count over the course of the cycle and came up with an increase of up to pretty much 3 additional heads per section for each one of the sections that we counted over here, which comprised 10 subsamples of 30 plants each. We also conducted foliar evaluations on the nutrient content. In particularly, I wanted to stop a little bit on the levels of microelements in particular. As you see on the right side of the slide, most of the microelements registered on the treated strategy with BioWorks strategy were able to register higher contents of microelements, and particularly iron was almost twice as much as the one registered in the non-treated control. And this could be related to the capacity that trichodermas have to issue siderophores, which are basically chelating agents for iron in particular. That renders even more easy for the plants to assimilate this particular element, and which is really important for photosynthesis, as you may know. Basically, the last example we had here was related to an image that showed what was visually present in those plants compared to the grower standard. As a side note, I should mention that the grower was able to obtain around $1,000 of revenue additionally when compared to the grower standard. So from all these experiments or trials, so to say, we can have two key takeaways. The first one is that Root Plus WP in combination with different biostimulants offers a viable solution to Fusarium wilt infections, both in conventional and organic crops, with a positive impact on yields. And not only that, we can see that the integration of Root Plus WP in crop protection programs can contribute to improving soil health, which is really important these days, as I mentioned before. With that, I wanted to thank you all for your time, and I would like to open it for questions.
Thanks, Eli. And we do have some. So, but just before that, I just want to remind everyone who responded to the attendance notification for the CEU credit, please do not close your browser before the webinar has ended completely, or you will not receive the email with the QR code. This is also for the viewers watching the archived webinar. All right. So for questions, okay. Root Shield contains beneficial fungal species. Are there any beneficial bacteria species that can also have similar functions?
Definitely, yes. There are a number of products in the market these days. Some of them contain Bacillus subtilis, some of them contain Pseudomonas, so Streptomyces. So the number will be pretty much really difficult to cover in just one presentation, but yeah, definitely the use of those other species can be combined with Trichoderma. I would say that Trichodermas tend to be a dominant species in many of the agroecosystems, so their use is essential, in my opinion, to combat many of these pathogens in particular. Fusarium, in particular, is very, very well controlled by these species of Trichoderma, so I would strongly recommend the use of Trichodermas for the combat of these pathogens.
Are these products available in Canada?
They are, definitely they are. We have registration of pretty much the same products that we have in US. They're also registered in Canada and in Mexico, by the way, if there is any person from Mexico.
How to use Trichoderma hazardinum, I know I said that wrong, for lettuce as it's only 20 to 45 day crop?
Yeah, it's a good question. We reinforce, one of the things that we recommend is reinforcing the use of Trichodermas, particularly during the seedling production. So basically, when you're producing those seedlings, those transplants, in a greenhouse, basically what we do is inoculating them as much as possible so that they have less transplant shock. And not only that, they're better prepared to overcome the conditions and the stress, in this case, biotic stress, by Fusarium, and also very frequently by Pythium, which is another pathogen that we find very frequently over here in California and in many other places, so to say.
Does the product efficacy depend on disease pressure and timing of the application?
Well, I would say to go on a preventative way, no matter what the level of pathogen that you encounter in your fields, you should probably go the preventative way. The use of, these biopesticides relies a lot on being preventative in their strategy. So inoculating the seeds would be the first step, if you have the possibility, of course. If not, inoculating the substrate of those seedlings as a second element, and this is really important. And the third one would be continuing on their field conditions. Depending on this crop cycle, you can do that either one time or several times, as we did in the celery or strawberry examples that we showed before.
Do you market your products in Colombia and Ecuador?
We have distribution in South America by one of our distributors, but I'm not sure if it is registered there. If they send us, we have a what we call technical questions channel available in our website. If they send us a message through that channel, probably we can send that to our distributor partners in Central America, which are the ones covering South America in particular.
What diseases were you monitoring for during the strawberry case study?
Fusarium, there are many, actually. Fusarium is one of them, but you can also find here other diseases like Macrophthalmia, Fusarina, Phytophthora cinnamomum, and Pythium. Those are very frequent over here in California, by the way, as a combined consortium of pathogens in this case.
Does trichoderma eat root exudates?
It does, yeah, definitely. And I don't know if I explained myself correctly during the presentation. I was a little bit nervous there. But basically, the plants issue a lot of exudates. And one of the functions is basically, feeding those beneficials that are present in the soil. Not only trichodermas, we can mention arbusculum mycorrhizae and other beneficials that might be the bacteria, of course. So that is essential. So what we want to do is not only potentiating the presence of those beneficials in the soil, but also potentiating the capacity of the plant to issue those exudates. And the way we can do that is by increasing photosynthesis in the plant. That's why we included foliar applications of amino acids, so that the plants is better suited to issue more exudates into the ground and feed on a better way those symbionts that might be present in the soil.
Do you recommend lab testing for levels of fungal disease or infection prior to application of BioWorks products, or simple assessment via visual cues by a professional?
No, having the criterion of a certified lab is always welcome. So if you happen to have that possibility, and both economically and technically speaking, that's even better, because you'll be more aware of the level of pathogens that are present in the soil and how you should probably structure the strategy in the crop. So, basically the answer is yes, because you'll be better prepared for what you have in the soil in terms of pathogen levels.
What was the increase in biomass of the romaine lettuce inoculated with Root Shield Plus?
Well, we didn't measure exactly in that case, the level of biomass we obtained. Unfortunately, we didn't capture that information, but we were able to increase the number of plants in the sections we measure. In particular, we actually counted literally the number of plants in 10 sections, and that was increased. And not only that, the level of biomass was increased, but we didn't unfortunately measure how much, if that is the question. Unfortunately, we didn't measure. But there was an evident increase by showing the picture I did show at the end.
Have you done research with Fusarium onion basil rot?
Can you repeat the name of the?
It says, have you done research with Fusarium onion basil rot?
Oh, not that in particular. We are working, if I understand well, because I don't know, probably I'm a little bit overwhelmed, but we are working in onions in particular. We have actually Dr. Michael Brambridge, if he's present, he could provide more information on that, but he has been working in onions in particular. I believe it was against Fusarium, their study in the Pacific Northwest. But we have evidence of efficacy of our trichodermas against pink rot in particular as well. So yeah, there are other pathogens that, although not within the Fusarium complex, are also a good target for these trichodermas in particular. Mm-hmm.
We apply trichoderma in a foliar spray.
Unfortunately, our products are not registered for that, at least in US. If it is in Canada, we do have a formulation that have been certified for that. The name is Ruchel HC, by the way.
How frequently should root shield applications be done after TP?
What is TP? Transplant?
Probably.
Well, I would say depending on the crop, you can do it, whether it is, in lettuce, I would recommend doing it at least once. In other crops that have, wider window, so to say, like celery, you can apply it up to 3 times after transplant. In strawberries, I would strongly recommend it as a dip, which is another alternative that you have, either for seedlings or unrooted material. And you can do it subsequently along the cycle of the crop. I wouldn't recommend stopping it because in most occasions you have a very high pressure, not only by Fusarium, but by other pathogens, as I mentioned before, Macrophomina, Pythium, Phytophthora. So you want to keep, if it is a long cycle crop, which is, normally stressed in some way by flowering and issuing fruits all the time, I would strongly recommend keeping the regimes of applications over the course of the crop. Probably stopping 1 month prior to finishing the crop, 1-2 months prior, but not less.
Do humic and fulvic acids help trichoderma work better?
They do. Actually, the product we combined it with both in celery and in lettuce, if you saw, is Chamay, which contains humic and fulvic acid. So the answer is yes. And the reason behind that, if I can expand a little bit on it, is that oftentimes we don't find the ideal conditions, especially in field production. We have a lot of, reduction of the organic matter content in the soil. And unfortunately, that leads to failure of the applications of beneficials oftentimes. So, we need to at least to have a carbon-nitrogen ratio, which is the one that guarantees a good establishment of trichodermas in particular, but also for other species, it's pretty much the same, although not at the same level. We have to ensure that the carbon-nitrogen ratio is the appropriate for trichoderma establishment. And if the levels of organic matter in the soil are low, then we are most probably going to fail in our effort. So the inclusion of this type of probiotics, so to say, guarantees that if the level of organic matter in the soil is low, at least the microbiome is accompanied with, a probiotic that would serve as the food source for that microbial, so that it establishes very well, especially at the earliest stages of the crop. And then the crop itself is the one that would, create that chain reaction by exudating, a number of metabolites in the soil, among them sugars, which are the one that feed trichodermas and other microbials.
If you apply other fungicides, like Postiva, for example, does that interfere with Root Shield WP's efficacy?
Well, off the top of my head, I cannot remember the active ingredient in that particular fungicide. But one of the technical elements that BioWorks is proud of is that we offer a very good compatibility testing system. And not only that, we have in our website, we have free of charge a tool which is called Compatibility Tool, and in which you can click over there and see if that particular fungicide is compatible with our product or not. So that is a free service that BioWorks offers, as I said before. And not only for Postiva, you'll find a number, actually, Ritual Plus has been tested against around, I would say, more than 400 pesticides in general, not only fungicides, but herbicides. You'll find fertilizers, insecticides that can be used as a soil treatment as well. So that's a tool I would strongly recommend to use by growers and other specialists.
Have you tested Root Shield Plus WP against Neopestalothopsis in field strawberries?
Actually, we have in the past, but we are testing it again at this moment as we talk. So, and again, this is a project that Dr. Michael Brownbridge has been leading. So he would be probably the go-to person to provide a better answer to that question. But the answer, in general terms, yes, we are testing it against it.
Can you use Root Shield in deep water culture?
Okay, so we're talking about hydroponic production. I can assume that. Well, if it is during the production per se, I would say it is not compatible in a relative way. And I used to produce hydroponic lettuce, by the way, in the past, while being in Canada. So I'm very familiar with that particular case. So what we used to do was reinforcing our strategy in the seedling production stage. That's what you want to do in those type of crops that unfortunately go or undergo deep water production system. The best you can do is inoculate those plants prior to transferring them to that deep water or pools, and reinforcing that strategy, you may probably reduce the amount of pathogen affecting the plants later on. Of course, there are more elements to that strategy, which include, disinfecting the pools on a frequent basis, minimum, I would say, every 6 months. And we do have in our portfolio other products that can be used in that sense, like Purpose Plus could be a product they can use in that particular case.
Are there commercial labs quantifying the level of fusarium in the soil?
Yeah, there must be commercial labs, both in Canada. Actually, in Canada, here I know there are some, and in Mexico. Yeah, it's a normal practice that growers normally recur to.
Does it make a difference whether you feed the trichoderma sugar such as blackstrap molasses to have a higher colony?
Yes, actually, the type of sugar that this type of microbials use for their both for their reproduction and spore germination and other stages in their growth is really important. Molasses, as you can see, is a byproduct of sugar production. Unfortunately, it's not a very how can I say, complex sugar. Its content is not a complex. It's basically not based on complex sugars, which are the ones that we prefer because if we use molasses, we risk of also increasing other pathogens populations. So the more sophisticated is, or the more complex is the sugar that we apply along with our products, and in this case, Chamay, as you saw, contains 5 sugars, but they're complex sugars. So with that, we guarantee that most of the use of those sugars is going to be basically done by Trichoderma and not the pathogen species that might be present in that agroecosystem, so to say.
All right, looks like we have time for one more.
Okay.
Can you provide additional details on why you suggest the combined treatments of biofertilizers along with trichoderma in your strategy? Isn't it enough to use Root Shield plus WP alone to combat the Fusarium complex and other pathogens in the field?
Yeah, again, as I said before, we oftentimes don't encounter the ideal conditions for Trichoderma establishment in the soils, especially in field production. This is a little bit more easy to find in greenhouse production, but in the field, we oftentimes don't find the levels of organic matter, and especially the carbon-nitrogen ratios. So by including the biostimulants, particularly the ones that I mentioned before, we have found that are very efficacious in that sense. You guarantee that carbon-nitrogen ratio is appropriate. So at the same time, they're going to guarantee a better establishment of trichoderma. And not only that, faster spore germination, faster and higher spore production. And that means that you have a better establishment around the rhizosphere and exclusion of those pathogens that might be present over there. Better results in general terms.
All right. I want to thank everyone for attending today's webinar. I especially want to thank BioWorks for presenting it, and a special thanks to our speaker, Dr. Eli Blanco Rodriguez, for providing valuable information about how biosynergies contribute to increased crop health and yield. If you want to watch the webinar on demand or revisit certain parts, it will be archived and available at the same link you used to access it today. You will also receive an email reminder of the archive with the link tomorrow. Thank you again for joining us. This concludes the webinar.